Good afternoon, everybody, and welcome to today's webinar on what is officially the first day of summer, although those of us joining from New South Wales will be forgiven for thinking that was a few days ago. My name is Ian Sheldrake. I'm a principal with the NOUS Group, and it gives me great pleasure to introduce the third in the series of webinars hosted by Dr. Norman Swan on the long-term implications of the COVID pandemic. These webinars have been organised by the WA Primary Health Alliance, are sponsored by the NOUS Group, and are raising money for a great cause in Youth Focus. I'd like to acknowledge that Norman and our other experts today have donated their time to participate. So on behalf of the WA Primary Health Alliance and Youth Focus, we'd like to thank you all for your generosity. I'd also like to acknowledge that we have people attending today from across Australia. So I'd like to acknowledge the traditional owners of the lands of this whole country and pay respects to elders past, present and emerging. The format for today is going to be a facilitated discussion, of which Dr. Norman Swan will be the facilitator. We'd like you to invite questions throughout, and you'll notice at the bottom there's a Q&A button and there's a chat button. The chat button is disabled, the chat function is disabled, but if you want to ask a question at any time, please do post it on the Q&A button and Norman will come to the questions at appropriate points in the conversation. So with that in mind, I'd like to hand over to Norman to introduce today's topic and our guests. Thank you very much.
Thanks, Ian. And it's great to be with you again, where we're looking at what will be the world's largest and fastest immunisation rollout in history. We're recording this, we're coming out live, but for those of you who are watching it in record mode, we're recording this on World AIDS Day. Another pandemic, which has been going now for 100 years or more, and but acutely for the last 40 years. And it's 80 million people have had it, 33 million have died, still no vaccine. And luckily with COVID-19, a respiratory virus, we've now got a plethora of vaccines, either just coming through phase 3 trials or about to enter phase 3 trials, certainly about to go to phase 1 trials. Well over 100 vaccines coming through. It's been an extraordinary moment in history, the history of science, medicine, and the control of a public health pandemic. And what we're going to talk about today are just some background information for you on the vaccines, what they do, what the state of clinical trials are, what we can believe or not believe from the press releases we've heard. Again, we're having this a little bit early because I think the first paper from the phase 3 studies will come out later this week, the week of the 1st of December in the Lancet. But it's not yet out, so we still don't really have a lot of visibility on some of these vaccines. And we're going to look at the practical planning that's going on at the Commonwealth, the thinking so far about how it's going to be implemented, what the options are, what you can expect for those of you who are watching from PHNs and primary healthcare sector in general and GPs. And Raj Verma is here from NOUS to talk about some of the systemic issues, both locally and internationally, that we might need to confront as we roll out vaccines in the current rather desperate environment. Luckily in Australia, we're not desperate because we've done the tough yards and we've got it under control, but at any time it could break out and we need the security of a vaccine as well. On our panel is Professor Terry Nolan, who is Emeritus Professor of Public Health at the University of Melbourne, is also at the Melbourne Children's Research Institute and the Doherty Institute for Infectious Diseases. And he was head of the School of Public Health at Melbourne University until recently. Professor Michael Kidd, sorry, hi Terry. Hi. Professor Michael Kidd, who has been on this webinar series before, who is Deputy Chief Medical Officer, Principal Medical Advisor, Department of Health and Ageing, Professor of Primary Care Reform at the Australian National University. Prior to that was Chair of the Primary Medicine Department at the University of Toronto. And talking about HIV/AIDS, Michael cut his teeth in general practice on being one of the pioneers of primary care and community-centered primary care for people living with HIV/AIDS, and has been a pioneer also in digital health as well. And Raj Verma, who is principal at NOUS, as I presaged, worked at very senior positions in New South Wales Health. So he's not the usual consultant, he's actually done stuff. And he's going to talk about the systemic issues to do with vaccines. Terry, let's get going, because Terry, the reason we're having Terry on is that Terry is an eminent clinical trialist with vaccines. He's not currently trialing in COVID-19, but that's what he spent most of his career doing, is trialing vaccines, particularly for children. Terry, just give us a kind of landscape of the vaccine landscape at the moment. We've booked 4. We'll come to the 4 that Australia's kind of put their shoe on at the moment, but just give us a sense of the landscape.
Sure, Norman. We are involved in a COVID vaccine trial, which we're about to start in the next couple of weeks. I'll tell you about that later, because that's another interesting dimension to your question about where we are. Just to cut through it, until about 2.5 or 3 weeks ago, we didn't really know for sure that we had a going proposition in terms of a workable vaccine. We had a lot of very good preclinical data, some non-human primate data, and some very good phase 1 and a little bit of phase 2 data, but the phase 2 studies have been accelerated into phase 3s. But it wasn't until the Pfizer...
you there, Terry, because
yep.
Not everybody will be across the jargon here. So phase 1 safety,
yep.
phase 2 is dose finding, and phase 3 is effectiveness.
That's right. So the Pfizer's announcement followed a few days later by Moderna's announcement. The 2 mRNA candidates were proof of principle. We knew that the target protein that all of the vaccines are directed at, which is the spike protein or components of the spike protein, because that's the protein that attaches the virus to the cell through the ACE2 receptor. So we had to be sure that it was going to actually work. And it showed not just that it worked, but frankly produced vaccine efficacy estimates way above what the most optimistic of us thought they were going to be, because it had been many years of trying to find vaccines against other coronaviruses, the ones associated with minor respiratory infections. And then when SARS appeared in 2003, attempts to find a vaccine before it was extinguished through public health intervention, those efforts also failed, although weren't complete. So it's not really true to say that we didn't have the technology. It just extincted itself, and therefore the pressure to find the vaccine was taken off. But here we have now a virus that we're very, very uncertain about, the possibility of safety issues, which we might come back to later, and also the fact that there were a whole lot of new platforms which were brought to bear on this particular virus that were effectively, in many cases, unproven. And so this news a few weeks ago was really spectacular, frankly. And then since then, just in the last few days, more data from a different platform, from the University of Oxford AstraZeneca vaccine, which is one of the ones that Australia has pre-purchased, with some controversial, but frankly, when you stand back and look at the data, still very, very good data, and some questions still to be resolved with that vaccine. And then last night, Moderna announced its final analysis on its 30,000 subject cohort, which produced another stunning definitive estimate of efficacy of the vaccine of 95 or 94.1%, but importantly, efficacy of 100% against severe disease. So again, great news, and we're very likely to have more good news in the next month or two from several other candidates that are a bit further back, but not far back in their phase 3 trials.
So the ones we're talking about here do the same thing, but in different ways. So you've got
yes.
2 mRNA vaccines, and you've got the Astra vaccine. So the mRNA takes the genetic message into the cell, tells the cell to produce the spike protein and generate the immune response. Astra does it by using a chimpanzee virus to take it in. And then the other 2, which Australia is interested in, is the Novavax, which is actually a more traditional vaccine where the actual protein is injected. You're not getting the cells to do anything, and you're inducing an immune response. And similarly with the University of Queensland.
Correct. The traditional vaccines, they were traditional only in the era of recombinant protein engineering, because the really traditional vaccines were just inactivated viruses. And some of the Chinese candidates are using that very old-fashioned approach. Again, when this all started, no one knew what was going to work. So some have gone with a very traditional route, and some have gone with the best new technology. But yes, you're right. There are different ways of delivering the vaccines, different levels of expectation about efficiency, about safety and so forth associated with each. So the full story on all of this is yet to be told. It's going to be a very exciting next few months as the results from some of the others come out. The detailed clinical trial reports, when they do appear in the scientific literature, rather than just press releases, which unfortunately at the moment is all we've got to go on. But no reason to believe that those press releases are in any way misleading, by the way. I think the companies are really, they don't stand to gain anything by falsely reporting findings from their own trials.
So let's just pick this apart a little bit. What do we know so far about safety? There was a halt to the Astra one because of this question of this woman with transverse myelitis. There might have been a second case of transverse myelitis. How sure are we that they're safe?
Yeah, there have been pauses, frankly, reassuring that the pauses have occurred because it tells you the system's watching. If those pauses hadn't occurred, and we'd found out about later, I would have been a lot more worried. Those two cases reportedly, and again, both for confidentiality reasons to protect the individuals concerned and also because of uncertainty about cause and effect relationships, two cases of possible some form of demyelinating disease. In vaccines in general, people think about chronic disease or disease where there is what's called mimicry. GPs will know about mimicry when it comes to autoimmune disease. If a foreign protein is introduced, which makes it look like one of the body's own proteins, an autoantibody response against that protein will generate an autoimmune disease. So if a vaccine somehow does that, produces a protein response that results in a mimicry with a consequent autoimmune cascade, that's a worry. So autoimmune diseases are very much in the top of the list of scrutiny for safety monitoring of all of these trials. And that's why that transverse myelitis thing was of particular interest. However, they were looked at very carefully to the extent that it's publicly known. The link, while it may have occurred, might only have been one case or maybe two. And in the aggregate, with the uncertainty of causality attribution, until more numbers are accumulated to see if there really is a signal there or not, does rely on the trial being completed. So in a sense, you've pretty much, unless it's absolutely certain, you've got a life-threatening, severe and predictable reaction to a vaccine. When you're dealing with studies with sample sizes of 30,000, you're going to want to complete the study and see if there is something going on there, whether the risk of that adverse event stacks up against the benefit of the potential harm that's prevented by the vaccine.
Now, you've got the health minister, the prime minister, and leaders around the world saying the vaccine's the answer. This is going to extinguish the epidemic. But that's not the basis of registration. The basis of registration is preventing the disease, which means that, let's say, 70% of Australians get immunized with a 90% effective vaccine. Yep. Then 50% of Australians won't get the disease, but 100% of Australians could still get COVID-19.
Absolutely.
And 40% still vulnerable. So the potential is the vaccine doesn't do anything apart from prevent the disease.
Well, that's right. And what we're interested in, that's why the vaccine efficacy of 100% against severe disease is something particularly notable. But the general principle that you've just articulated, which is the fundamental of licensure, is an overall favorable risk-benefit equation. If the intervention, whether it's a drug or a vaccine or anything else, generally is going to do much more benefit than cause harm, that's the basis for licensure. Licensure doesn't require a vaccine efficacy of 95% compared to, say, 75%. 75% is a good number, frankly. And if we hadn't had these other responses from these other products so far that look like they have a very high efficacy, that might have been the best that we could have imagined we'd get. So the full story hasn't been told yet, and it is quite possible that vaccines that will have more modest efficacy will still survive licensure and still be regarded as beneficial, and particularly in the absence of adequate supplies of alternate vaccines, which may have higher efficacy in an urgent situation, there may well be a place for something that's not quite as optimal as the perfect solution.
My point is, when will we know, how will we know, given that the trials haven't always been designed to pick up infection control, when will we know whether they will prevent transmission in the way that measles immunization prevents transmission?
Yep. So one or two of the studies are actually doing infection surveillance. So the Oxford AstraZeneca is one of those where they're actually testing not on the basis of symptoms every week, but just as a routine. So in terms of being able to pick up infection that may be subclinical. So for some of the trials which are doing that as part of their protocol, that's a massive exercise, I should say, by the way. These trials are big enough as they are, but putting that on top of them is a big ask. But the second way will be over time in looking at whether there is a visible herd effect, in other words, reduction of disease that might be attributable not to being directly immunized, but those around you who've been immunized. So there are indirect epidemiologic methods to actually assess that. And so they're obviously much less precise, and it would be much better to have a more direct estimate of this prevention of infection versus prevention of disease issue.
So first of all, it's an act of prevention in terms of disease, which is good.
Yeah.
We're gonna have to wait and see whether it actually extinguishes the pandemic.
So the best idea we have about prevention of infection so far before the phase 3s and all the complexity I've just described is actually from non-human primate challenge studies. So these are macaques, generally monkeys, which have been given virus after being immunized. And I've just reviewed all of the non-human primate data from all of the major candidates, and none of them was able to completely sterilize virus from the nose after a macaque had been initially vaccinated and then challenged with live virus. Every single macaque that had been challenged in every single study had survived and did not have any significant pneumonia. So we knew from the monkey studies that it was very likely that very substantial protection from all of these vaccines that have been tested in this way would produce. The problem with virus in the nose is that you also got virus which you're putting in the nose. So the inoculation for these challenge studies, about half the virus gets inoculated into the nose of the monkey and the other half down into the trachea and into the lung. Having residual virus from that challenge can also then be picked up when you're looking at whether or not the vaccine has prevented the virus from becoming established. There are some subgenomic analyses which will allow you to make that distinction. And by and large, in those studies where they were done, the subgenomic analysis showed a more favorable response. In other words, there was a little bit of probable of infecting virus, which was still present, but some which was actually replicating virus, indicating that the vaccine hadn't produced in the nose. Remember, this virus is a mucosal infection initially, and that's why it's so important to have mucosal protection. And we're not using mucosal vaccines. We're using vaccines which are given systemically. And therefore, it's a bit of a long ask or a hard ask for these vaccines to protect mucosally as well as systemically, hence your question. By and large, though, it's quite possible we are going to get a surprise on this and have in humans a better level of infection prevention than we might have expected. I know that the Chief Scientific Officer of Moderna said exactly this in an interview a couple of days ago, that he's quite suspicious that it's going to turn out much better than we had hoped.
Well, that's great news. We'll come back to you later, Terry.
Yep.
Michael, thanks for joining us again. Just give us a sense of, so you've got 4 vaccines that the government's got arrangements with. University of Queensland, which is furthest away because it's only about to start its phase 3 trial. Novavax, which is a protein-based one, which the phase 3 will probably report January, February. Pfizer, which is the mRNA, which has just reported and asked for emergency authorization in, well, has it? Yes, I think they have, in the United States. And the Astra vaccine, which has a little bit of controversy around it, but which we're manufacturing at CSL. So give us I mean, obviously you haven't, well, maybe you have tied it down what you're going to do, but just give us a broad view of the thinking so far, priority groups, phasing, because some people are worried that if you mess around and you don't vaccinate enough people quickly enough, that the virus could mutate around the vaccine. So there's a delicate balance here. What's the thinking?
So the Australian government and the National Cabinet released the Australian COVID-19 vaccine policy coming out of the last National Cabinet meeting on the 13th of November. And that outlines the current thinking about the rollout of the vaccine, including the the preliminary priority populations, because of course this may change over time, thinking about logistics, thinking about the locations, the sites where the vaccines will be involved. And obviously for general practice, for primary care, people are very keen to know now whether they're going to be involved or how they're going to be involved in what you've described rightly, Norman, as the largest vaccination program in human history. So the preliminary priority populations, there are three. The first is those people who are most likely to be exposed to COVID-19 in Australia, and this includes healthcare workers, people working in aged care, and perhaps also people working in the quarantine settings, given that most of our cases now are in people who are coming back to Australia from countries where the pandemic is much more active and out of control in many of those countries. The second priority population is those people who are most at risk if they were to contract COVID-19. And obviously that includes older people, particularly residential aged care facility residents. It includes Aboriginal and Torres Strait Islander people. It includes people with certain chronic health conditions, people who may be immune suppressed. And the third group is people who are providing essential services, so that if we did get another very serious outbreak of COVID-19 in one of the states or territories or across the country, the people working in the areas where we need those people to continue to work, and otherwise the nation grinds to a halt. So they're the current three priority populations, but as I say, that may change over time. The actual availability of the vaccine is still to be determined. Two of the vaccines will have the capacity to produce onshore in Australia. Two of the others will be reliant on those vaccines coming into the country, having been produced elsewhere. Clearly, even though the Australian government has contracts for these four vaccines, there are other countries which have contracts for those vaccines as well. So how quickly we will see large numbers, large doses of those vaccines available in Australia is still remains to be seen.
And just to be clear, it's the University of Queensland vaccine and the Astra vaccine that we'd make locally through CSL. The other two would be made overseas, which is Novavax and there. How soon do you expect TGA approval for, say, the Pfizer and the Astra vaccines, which are furthest on?
Well, it's I don't know exactly the answer, but the projections at the moment are probably at the end of the first quarter of next year or into the second quarter of next year. In part, it depends, as Terry is describing, on how the trials continue, the reporting. We're yet to see the peer-reviewed reporting of these trials and what happens with those vaccines as they go through the TGA process of being deemed to be safe, effective, and appropriate for use in Australia.
I mean, you're not a politician, but it creates a political problem if the FDA and the British agency give regulatory approval this month, and we're 3 months behind, does it not?
It may or it may not. We have our own processes in place to ensure the safety of vaccines, and we follow through those processes.
So let's just, and then you've got the general population which follow on from those priority groups. And let me clarify, unlike the British government, which has already said they're not going to immunise the whole population, our intention is to do that, is it not?
Certainly the contracts which we have provide enough doses of the vaccine to be able to vaccinate the entire population of Australia. Most of these vaccines, which are currently going through the clinical trials, require 2 doses of the vaccine. So whatever number you see, we need to double for the population. Right?
So how are we going to do it? You've got one vaccine that requires transportation at -70. the other's fridge temperature. So how are we going to do this? And who's going to, particularly as you get into the really big phase, which is, as you've described it, it's really a telescope, smaller group, slightly bigger group, much bigger group, and then a huge group, which is like 25 million people. Who's going to do it? Where? Given that you've got a cold chain that's much more critical than we've ever experienced before with the Pfizer vaccine.
So the Australian government will be responsible for distributing the vaccine out to the sites where vaccination will take place, and will also take responsibility for making sure that the cold chain is maintained for whichever of the vaccines we're distributing at a particular time. Obviously, there's going to be close work with each of the states and territories in working out how to get the vaccine to the populations in each of those states and territories. Now, at the moment, there are a number of locations for vaccination which have been identified. Clearly, the site of most immunisation in Australia is through general practice. And so general practice is there at the top of the list. The Australian government during the pandemic has established 150 general practice-led respiratory clinics covering both metropolitan, rural areas and in many Aboriginal community-controlled health organisations. These clinics may also end up being sites for the immunisation program. Each of the states and territories has their own vaccination centres. The important thing is that drive-through options will probably not be appropriate, because we will need to have the opportunity to observe people after they've been immunised to make sure that we're picking up if there are any immediate adverse events occurring from the vaccines. We also have to look at workplaces. Clearly, the most efficient way to immunise the people working in hospitals, in general practice clinics, in aged care facilities may well be in the workplace themselves, which of course already happens with influenza immunisation each year in Australia. And then we have to look at the in-reach. How do we go into aged care facilities and immunise all the residents, immunise all the people working in those facilities, and the same with Aboriginal and Torres Strait Islander settings, going into communities both in urban, rural and remote areas to immunise the population. So there's a lot of work underway. Community pharmacy may be involved, but it's being looked at as a possibility probably once things are well established and we understand the safety profiles of the vaccines afterwards. We will be looking at recording all the vaccines in the Australian Immunisation Register. And in part, that's to follow up and make sure that when people are getting their second vaccine, they're getting a second dose of the same vaccine. We don't want to risk people getting two doses of different vaccines. We don't know what that's going to lead to with regard to the efficacy of the process, but it's likely to be suboptimal. And we need to be monitoring for side effects as part of the post-marketing surveillance, if you like.
Phase 4. And just briefly, before I go to Raj, the role of PHNs in this?
So PHNs will likely be involved. Again, it may depend on what happens in each of the individual states and territories. So the Commonwealth will be working with each state and territory. We did see a wonderful program with the PHNs this year to ensure that every resident of every residential aged care facility in Australia was immunised against influenza. And the PHNs reached out to each residential aged care facility, found out whether they were had arrangements with their local GPs who were providing services to that facility to get everyone immunised. And if they hadn't got that arranged, or if the GPs who normally did that were themselves vulnerable people and were having to be in isolation, arranged with other general practices to get that vaccination program happening. So there may well be a range of ways in which PHNs are going to be involved in the program.
Raj, you've identified various risks that need to be taken into account. Your microphone's still muted if you want to unmute. You've looked at manufacturing and deployment as a risk area. vaccine hesitancy in the general community,
Yep.
and the epidemic itself. Do you want to just run through that and how you see those risks and how they
Yeah, so I think, thanks, Norman. In fact, I'm confident and optimistic that we will get this right. But I also think that the people who are usually doing vaccination and work in the vaccination system may not have taken into account the pressure and the some of the things that will come up. For instance, if the deployment is delayed, there will be a black market effect created where people will want to buy their way to the front of the queue. And so they will find ways, and they have in many other markets where they find a way to procure or steal or whatever else the vaccine, so they and their friends and family can get it first. So issues of security will need to be thought about, which is not something that anyone would normally be thinking about in a vaccination program. So that, you know, there are issues around that. There also, and I'm really pleased that Michael is pushing the immunisation register as a way to track, but we have had a very poor history of compliance with that immunisation register and making sure that the information gets into My Health Record, because there will be a strong demand for a passport, if you like. I've been immunised, here's my proof. Now, it is in My Health Record if you go through the immunisation register, but it's many steps, remembering passwords, having the app, having a passcode to get into that. And, you know, you have businesses, Qantas last week or the week before said, we're going to want people to prove that you've been vaccinated before you can fly. And I don't know whether the government can stop that sort of move happening, but there'll be employers and workplaces and nursing homes and all sorts of people trying to put up that barrier of you can't come in until you can prove you've been vaccinated. So an easy to deploy passport of some description or app would be really helpful. In terms of the vaccine hesitancy, jumping on any sort of adverse event and being able to manage the message will be important, because the media will be all over. They'll be at the front door of the first person who's had some sort of adverse event. And even if there's 1 in a million serious adverse event, that's 25 people in Australia, and I don't know how many 100,000 around the world who could, you know, have significant adverse events. And then how does that impact people's willingness to be vaccinated? Because the hyper pressure and view from the public and the media around this will escalate very quickly. And, you know, there's already the anti-vaxxer and you know, you look at just masks being politicized. So I've no doubt that people will attempt to make the vaccination a political issue. So how we get media people like you, Norman, doing your calm messaging, but having other people, you know, getting to other audiences to manage this message will be really important in terms of trust in the vaccine and the responses. And around the severity and managing the pandemic in the interim will be important, because people will have a sense of, oh, the vaccine's happening, I can relax, I can go back to the old ways, and social distancing can disappear. There will be businesses pushing to do that pretty much straight away. And so who's going to set the threshold? Is it 20% of the population is vaccinated and we can open up, or do we have to wait till 90% are vaccinated and then we can open up? And who sets that number? And, you know, they're the sort of arguments that will come forward that you need to be prepared for, that may not be, you know, in the normal vaccination course of events, things that have been thought about.
Thanks, Raj. Terry, what do you think about the vaccine hesitancy in the community? I mean, Australians by and large have been pretty good at getting immunised.
Well, we weren't that good when H1N1 happened. The coverage we got with the pandemic vaccine in 2009 was around 30%. So I am very keen to see, as Michael's just reported, that we take this very seriously and not anticipate it's going to be a breeze. It's a 2-dose vaccine. I noticed, Michael, and thank you for pointing me to it, I hadn't seen the advice from the National Cabinet, but hidden at the end, not hidden at the end, but at the end is what looks like a non-concluded remark on whether incentives might be built into the promotion structure for ensuring people do get their second dose. So of all these candidates that are now we're waiting for, only one is a single dose candidate, and that's from Johnson & Johnson or Janssen. it's another adenovirus vector vaccine. And most people are pretty skeptical that one dose will be enough. Most of the trials won't have data on one dose protection because the compliance with getting the full 2 doses in the trials will have been very high. So we won't really know for sure the extent to which one dose does translate into some level of vaccine efficacy. And we have to assume that it's pretty hopeless without it. That's certainly what the antibody studies that led to the 2-dose regimes suggested for all of the manufacturers except one, as I mentioned. So I think I'm pleased to see that there are many creative opportunities being considered. I personally think that an assertive strategy, which is impatient to get the vaccine out as quickly as the suppliers will allow it, we should pursue rather than have a relaxed attitude about waiting several months to just let it all happen. The Americans have announced very, very ambitious logistic arrangements to get the vaccines out if they get emergency use approvals within a day of that approval to the distribution centres in each of their 50 states. And then within a day of reaching the distribution centres to be able to distribute using DHL and FedEx and so forth. They are going to approach this on a massive scale. I'm not saying military scale, Michael. The parallels with using the military have been troublesome with the whole COVID response. But being very organised, very energetic and very ambitious to do this very, very strongly, because it is a very, very big ask, as you said right at the beginning, Norman.
So why was it only 30% in 2009?
Probably the same as I noticed in the chat. A couple of people are asking the question, and I've heard this too, that, and this is particularly the case in parents of children, they think the disease is not nearly as bad as the risks of the vaccine. So particularly for young people, very, very low case fatality rates and low rates of severe disease, albeit not zero, but nonetheless very, very low. And still with uncertainty about safety in the longer term, and we're all going to have to wait and see what that happens, despite our reassurances in the interim and the thoroughness of the safety monitoring that's happened within the trials. I think that's the main reason. And at the time of H1N1, there was a view that it was a Clayton's pandemic. We had all prepared and were involved in H5N1 planning for our pandemic plan at the time. This is when I was involved with ATAGI. And basically looking at case fatality rates of up to 50% were the worst case scenarios we were looking at. And then along came H1N1 with very, very low overall population fatality rates and a degree of cynicism, including from the medical profession, that this was really not that serious and therefore not really worth worrying about too much. Of course, that was ridiculous when you looked at the population morbidity and mortality data. The effect on pregnant women, for example, was staggering, and also elsewhere in the population. This is a virus which affects the elderly preferentially as well. H1N1 affected young preferentially. The elderly were protected against H1N1, and we didn't see anything like the severe disease we've seen in residential aged care, for example, and so forth, where it's just been disastrous when it's been allowed to disseminate.
One question here is, to you, Michael, is it realistic to immunise remote communities with a vaccine that needs to be stored at well below sub-zero?
I think many people will have seen John Skerritt from the TGA talking about the sophisticated eskies which are being developed to deliver that vaccine and to keep it at the very low temperatures, and so that technology is being developed at the same time, and we'll roll things out. Again, it depends on which vaccine or vaccines become available first, as to what the cold chain requirements will be in different settings. I also just want to touch on the issue about the community acceptance. This year we saw record numbers of people getting vaccinated against influenza, and the concern that people had about the potential double whammy of having COVID-19 and influenza happening at the same time, and the impact that may have on our on our healthcare system. We distributed 4 million more doses of the influenza vaccine this year than we did the year before. So I think there is a degree of vaccine readiness, but I think that Terry's right. The communication is going to be absolutely essential, and keeping on the front foot with keeping people informed about whichever vaccine becomes available first, so that people are well informed about what's happening, do understand what the priority populations are, do understand how they will get access to the vaccine, do understand the safety profiles and what's being done to monitor for adverse events to make sure that we are doing all we can to protect the population.
The next one I ask of you, Terry, and I'm paraphrasing a question that's up here on the screen, is, but I get it a lot on CoronaCast, which is, how will we know, or is there an expectation that some vaccines will work better in some groups than others? I mean, we're already designing influenza vaccines for children and older people.
Good question. And the FDA in particular was very aware of that issue in providing its guidance in the design of the clinical trials and requiring the manufacturers that did the phase 3 or are doing the phase 3s to be sure they had large enough groups of particular at-risk populations. So that's the elderly in particular. Most of the phase 3s have been designed with at least 25 or 30% aged over 60 in their populations for that reason.
And comorbidity.
And having comorbidities as well. And also then people of different degrees of social vulnerability. So people from different racial and ethnic backgrounds in the studies as well. Moderna, I know, colleagues at Moderna told me they were pulled up by the FDA as they were recruiting because they weren't happy that they had enough individuals from African American or Latinx background and so forth in their population. So they had to really strive to increase the rates of accrual of recruits for their studies to beat the FDA's requirements. Down the track, are there going to be specific biologic characteristics of these different platform vaccines that might mean that there'll be either different side effect profiles or different benefit ratios in different populations? They're possible. At the moment, it's not visible that that'll be the case, but it's too early because we don't yet have the detailed data on enough of those populations to look at those subgroup analyses.
Another question here, I'll also go to you, Terry, is, and again, I'll paraphrase this, it's about long-term side effects for a specific vaccine, but I'll roll into this another, that's one question. The other question is, and just to go to the technicality of the Astra vaccine, the viral vaccines, the adenovirus vaccines, is that it's been well publicized that the half dose followed by the full dose looks as if it gets a higher effect, reportedly 90%. The theory is that it's not induced antibodies to the chimpanzee virus, and therefore the second dose is more effective when it's a double dose. The Russians get around this by giving 2 separate viral vectors, so you don't get the same one and the second one. So first question is, if that's true, will you be, and you need, let's say you need annual boosters, which we don't know yet for these vaccines, will Astra be cut out of the annual boosters because of the very nature of it, that you're actually immunizing against the vaccine? That's the first.
Yep. Okay, that's a good one. That you've stacked 3 hypotheses, one on top of another there, Norman. So
we'll come back to this.
No. I'm not being frivolous here. It's a great question and a very hard one to answer, especially the one with the adenovirus. Oxford chose the chimpanzee because it wasn't expected there would be antibodies to chimpanzee in humans. They are measuring that now, and in their response to last week's results, they're going to go back and have a look much more carefully at just what's going on. The hypothesis you just mentioned was one of several which has been proposed, all credible, that might explain the unusual, but frankly, for a vaccinologist, not actually drastic revelation that less is better. It's not at all unusual in that we're not talking about drugs here with very, very simple dose response curves. Vaccines are biologics. They have complex impacts on B and T cell function, and that are not necessarily dose dependent at all. And so it remains to be seen how that's going to pan out. So to come to your question down the track, the second hypothesis, if that were the case, would it be the case that potentially boosters for an adenovirus vector vaccine, human or chimp, irrespective. That's possible, but we're in such a good situation because we're going to have a whole portfolio of very good vaccines. So frankly, if we had to use a different vaccine for a reason like that, we're going to be in a pretty good place.
How are the second wave? They've been able to test these vaccines super quickly because there's so many people being infected with COVID-19.
Yeah.
They're able to do these in unbelievably fast time. And even if Astra's got to go back and do a third trial, they're probably going to be able to do that incredibly quickly too.
Absolutely.
But how's the UQ going to manage, or second time around when there might not be that much COVID-19 around? How do you test second generation vaccines?
Well, I should disclose I'm on the advisory committee for the UQ phase 2/3 study, and so this is a good question. And if ready availability of licensed and highly effective vaccines do appear, it will be probably impossible, certainly challenging ethically, to do a placebo-controlled study with a vaccine which is further down the track. So I'm involved at the Doherty with colleagues involved with what we hope will be second generation vaccines for various reasons, likely to be even better potentially than what we've got now for various reasons. But this is an issue for us as well. Relative efficacy studies are common now. We do them with flu all the time because it's unethical where flu is not just recommended, but also funded in populations to give placebo and ask people to forego the benefit of a recommended intervention. And so giving one vaccine against another, it is possible to estimate the equivalence, if you like, the non-inferiority of one vaccine against another. The problem with that is it increases the sample size of the studies and sometimes their complexity. But there are ways out of this. We don't have to have a situation where we have to have a placebo comparison. And then the final thing is, if a correlate of protection emerges from the existing studies, we were talking before about surveillance for prevention of infection, but if a blood test result, an antibody response, which allows you to link your response to a vaccine to give you a high probability that you will be protected, that's the correlate of protection, then we'll be able to infer protection and not have to do an outcome study. We can just measure blood antibody levels.
Question for you, Michael, is just on distribution. Is it all going to be through mass vaccine sites? Have you got down to that level of logistical planning yet?
Well, certainly what we know is that the vaccines coming through are multi-dose vials. So you need to be able to immunise a certain number of people to make sure that we're not getting wastage with the provision of the vaccine. And obviously we can't afford to build in wastage, especially in the early stages of rolling out the vaccines. So it will be likely that the vaccines will be in places where significant numbers of people in those initial priority populations can come together and get the vaccine at the one time.
Oh, so we're going to have so that's an inherently risky, that adds a level of risk, doesn't it, a multi-dose vial?
It does. I mean, Terry might like to talk on it as well, but clearly there are additional concerns about the safety because of the multi-dose arrangements, and it'll be absolutely essential that all the people who are involved in administering the vaccine have been appropriately briefed, educated, trained in making sure that the vaccine is being used as it's intended and the safety provisions are being addressed.
And we've done it before, Michael. You'll remember in 2009, multi-dose vials were used. We had to deal with the thiamersyl issue, the preservative, which is often used in multi-dose vials, and people's concerns about a mercury-related compound in them. We had to deal with issues of people concerned about transmitting infection and so forth as a result. It was dealt with effectively because there were the same questions, Norman, from healthcare providers as well as from, including specialists, by the way, not just more generally, who had their own particular prejudices about multi-dose versus unit dose preparations. There's no question in a pandemic situation, there's just no, you've got to use multi-dose vials. The logistics and issues around the vaccine itself are bad enough, but the logistics around enough glass, enough stoppers, and all the rest of it are horrendous. And particularly if, as I was saying before, if we move to do this at scale and do it quickly, it makes no sense to have all of this massive throwaway plastic when in fact you've got people queued up ready to get their vaccines from a multi-dose vial.
I didn't get you to answer the long-term side effect question because I was asking such a complicated question. First up,
thanks for forgetting it.
How long with new platform technologies would you have to watch before you can start relaxing that you've not got a major problem in your hand?
So we omitted to talk about the single most important theoretical concern for these new vaccines against coronavirus is what's called vaccine-associated enhanced disease, which was seen in vaccines against respiratory syncytial virus, RSV, in the late 60s in babies when a small number of babies actually died having been vaccinated when they were then exposed in the normal course of life to a natural RSV infection. Got just such a severe reaction, it killed them. In a way, it's a parallel phenomenon to what happens with dengue, with second infections with dengue, when you're exposed to a different serotype or genotype of dengue than you might have been initially. And really the full reason for this is not fully understood, but although much better understood now than before, it's probably to do with the first vaccination producing antibody that is not as direct as it should be against the virus. And as a result, when you're subsequently exposed to the virus, the immune system is fooled into thinking that there is still an ongoing viral challenge when in fact it might have been dealt with. And if you like, the host response, the immune response is so severe that it causes the damage. So that's why I mentioned before the non-human primate or the monkey studies which were done were because people were very concerned that might be the case with this coronavirus. It had appeared in some of the early studies that were done with SARS in 2003-04 when the vaccine was being looked for there, and a hint that this virus might actually do that as well. In fact, there's been such careful scrutiny of all of the immunologic predictors of this type of reaction occurring, what's called Th1 versus Th2 imbalance in responses, more of a balanced T cell response rather than just a very profuse and imperfect B cell response, that there has been no signal in any of the candidates that might be the case. And when this reaction occurs, when you've got a lot of circulating disease, it's likely to occur within a couple of months of being vaccinated. And that's why the FDA has required a minimum of a median of the enrolled populations to have had 2 months observation after the second dose of their vaccine in the trials, because that's the period that is thought to be, if it's going to occur, that's when it's going to occur. And it has not occurred so far. And all the animal and other data so far as well suggest that's not going to happen. Any other longer term potential effect is the same with any drug or any vaccine. Of course, it's a possibility. It'd be foolish to say that, you know, nothing could ever happen. You can never say that with certainty at the onset. So having very, very strong pharmacovigilance in place, as Michael was suggesting before, is in that document, is absolutely essential. And it has to be more than just passive surveillance. There has to be a level of active surveillance to really be sure you're capturing any cases that are occurring down the track.
Michael, let's hope that the vaccines do prevent transmission rather than just disease. If they were to prevent transmission, what does the government calculate is the herd immunity required? The number 60% has been bandied around. Is that what the thinking still is, that if these vaccines are effective at preventing infection, you've got to get to 60% of Australians?
I think there's still work to be done to work out exactly what figure we should be landing on.
Right. Okay, we'll just leave it with that. And another question here, what research is being done in the communication sphere to know what messages, I know you've got to go in a second, I apologize, to the research into communications to know what messaging you need to convey, coming to Raj's point?
Yeah, look, there's a lot of work underway looking at the messaging which needs to come across. Raj identified a number of the important concerns which people may have about the vaccines. I think this is really important, especially for the general practitioners online. Many of our patients will be asking, as the vaccines look like they're starting to become available, they'll have a whole series of questions about when will it be us, how do we get it, What are the side effects? Is it going to be safe? Which one, if we've got a selection of vaccines, which one would you recommend for us? Has this been shown to be beneficial in elderly people? Has this been shown to be safe in children? The list of questions goes on. So communication strategy needs to be very sophisticated, because this is going to be something which we are all going to be talking about for much of the early part of the coming year.
I know you've got to go, Michael. Thank you very much for joining us.
Thank you, everybody.
So, Raj, there's been a long conversation here. What comments have you got just on the topics that we've covered so far?
Yeah, I think it's, and due respect to Terry, the vaccine efficacy is going to be important, but a couple of Harvard and Yale public health professors last week did a lot of mathematical modelling, and I'll quote, "Factors related to implementation will contribute more to the success of vaccination programs than a vaccine's efficacy as determined in clinical trials." So we got to trust the scientists. I'm a scientist. I worked as a laboratory scientist for many years. So you got to trust the scientists, but there's a lot of implementation work which will make or break whether or not we get to fast immunity in the community so that we can resume business as usual. So.
And Terry, despite the fact I was giving Michael a bit of a hard time on the approval side, there might not be a bad thing for us to sit back a little bit and let other people make mistakes.
And I would have said to Michael, a better answer, Michael, rather than trying to carry it for them, is to say they're working as hard as they can to deliver it as soon as they can, with plenty of encouragement from us. So I thought it was a very good question. And the TGA is not that different from the FDA or EMA or MHRA in the UK. They are talking to each other all the time anyway. Normally, not let alone now during COVID, they will find ways to accelerate their appraisal. They're all looking at the same data. It's not as if they're looking at different data sets. So there's no reason why it has to take a long time unless there are deficits in the data. So for example, if the AstraZeneca issue turns up as being something that they really feel is a deal breaker for giving a provisional license, then and they say we can't really make that decision until we see the data, well then the clock stops. They have to go get the data and come back. So you can't hurry that up. But in terms of all the data they need are there, then the process of examining those data and so on, by the way, it doesn't require peer-reviewed publication. I mean, that's my mantra. I'm a scientist and I'm as critical as anyone else of trying to make these judgments just on media releases, but the regulators don't have to have peer-reviewed papers. Most of what they see is not published in peer-reviewed literature either, because it's too early. The companies have done the studies and they've got very, very detailed dossiers, which are far more detailed than a short 10-page or 5-page paper in the New England Journal, a lot more detail that they have access to. And they can talk to the company, they can quiz them about the data, they can send them questions and so forth. So that's another, it's sort of a furphy in terms of another delay in terms of them needing to see the peer-reviewed data.
Look, thank you very much, Terry. Thank you, Raj.
Pleasure.
Really fascinating. Lots of questions as well. And I'll hand back to Ian.
Thank you very much, Norman, and thank you to Raj, Michael and Terry for contribution. I'm sure everyone would agree we could have carried on for another couple of hours on this topic and barely have scratched the surface. Before closing, I would like to remind everybody that this is a series of webinars and this is the third of 5. If you have signed up for a season pass to all 5 webinars and you've yet to be able to access your recording to the first 2, please do follow up on that. There were 2 fascinating conversations over the last couple of weeks. And just to remind everybody, we have 2 webinars next week, both on Tuesday and Wednesday. Tuesday's webinar will be focusing on the long-term impact on the nation's mental health, both through the vaccination program and in the aftermath of the pandemic. And then the Wednesday conversation will be focused on the impact on Australia's children, and in particular the sort of the 8 to 14 year age range. There's been a lot published around the economic impact of school leavers and the ability to kind of find their way into employment. But we're going to focus in particular on those young people in the country who are aware of what's going on, but maybe don't understand or unable to fully process it. So we've got some great guests and we hope you can join us. So thank you, everybody. And we look forward to seeing you next week.